All right, this video is going to be all the steps of cellular respiration, aerobic cellular respiration. It's going to be a little bit long because I don't want to break it up. I'd like for it to all be condensed so you can find things more easily. During cellular respiration, the main goal is to break down our foods and get energy out of them. That energy is stored in the chemical bonds between them and between the molecules, the bonds, and it's all about those electrons being transferred around like we talked about in the last video. So this is going to be showing you an example of we have sugar, and that's going to be our main molecule that we talk about during cellular respiration. In another video, we'll talk about how other different macromolecules can be used to generate energy. So we have sugar here. It's a 6-carbon molecule, and the idea is that we can break apart those carbons to get the energy from between them, and in the process what happens is that we end up making carbon dioxide, water. Those are byproducts, and the big takeaway is that we're going to make a lot of ATP. So you can summarize the equation here as C6H12O6, that's the chemical formula for glucose, plus 6 oxygen. You have to have oxygen for aerobic cellular respiration to occur, yields 6 carbon dioxides, 6 waters, and a whole lot of energy. So when you're breathing in oxygen, that oxygen is getting into your blood, getting into your body, into your cells, particularly into your mitochondria, and it's going to be utilized in the process of breaking down sugars. When you exhale, that's the excess carbon dioxide that has been created in the process of cellular respiration. When you breathe out, you know how if you put breath on glass, it gets droplets on it because water is one of the things that comes out as well. And then you've got the energy to help you move and do all your cellular functions. So there are 3 main stages to cellular respiration: glycolysis, the citric acid cycle, which you will often see called the Krebs cycle because the guy who first described it was named Krebs, and oxidative phosphorylation. There's also a transitional section here called the oxidation of pyruvate. I have a diagram here that is more generalized, and it shows how glycolysis happens. This is a whole cell. Glycolysis happens in the cytoplasm. And then inside of our mitochondria is where we're going to have the Krebs cycle and oxidative phosphorylation. So the first step is glycolysis. Glycolysis literally translates to sugar, glycolysis, splitting, glycolysis. It's a universal process found in all different types of cells, and it occurs in the cytoplasm. Since all cells have cytoplasm, and since all cells do glycolysis, scientists think that this is the very first metabolic pathway that evolved. So the idea is that we start with a glucose, a 6-carbon glucose over here, and we break it apart into 2 3-carbon molecules and create a product called pyruvate. So we are breaking apart a 6-carbon molecule, and we also are partially oxidizing it. Remember, oxidation is the loss of electrons. The products here, 2 pyruvate, 4 ATP, although there'll only be 2 net ATP, we'll get to that in a minute, and 2 NADH. These NADH are electron carriers. They get reduced, NAD plus gets reduced, and this NADH is then used in later steps of cellular respiration. So this step is really important for generating just a couple of ATPs, but important for generating these electron carriers that will get moved into a different section of cellular respiration. The first half of glycolysis is what we call our energy requiring steps. So recall in the last slide I said that there were a net of 2 ATP, and that's because in this step we actually have to use 2 ATP first in order to get this process started. So the first phosphate is added by hexokinase. Remember, kinases are things that phosphorylate molecules. So kinases are enzymes that phosphorylate molecules. So here we've got a glucose that is going to be converted into a glucose 6-phosphate. And so what happens is we use an ATP, hexokinase removes that phosphate off of the ATP and puts it onto glucose. That ATP then is generated and it's made into ADP because one of the phosphates is removed. The second phosphate is added to that fructose 6-phosphate after it does some other stuff. You don't have to memorize all of these steps. I'm just talking to you about what happens. And a second phosphate is added by a different kinase, and then we end up with this 6-carbon molecule that has 2 phosphates attached. So that glucose gets split into, the original glucose gets split into 2 carbon molecules, and each of them have a phosphate on them. So we've used 2 ATPs in that process. Then the second half of glycolysis is an energy releasing step. So big important takeaway here is that 2 times, see over here it says 2 times, it's going to happen 2 times because we made 2 of those 3 carbon molecules, 3 carbon glucose derivatives. We have 2 times happening that we're going to reduce NAD plus into NADH. That's going to be a big limiting factor because if we cannot if we cannot be making more NADH, we won't be able to then put it through to the later steps of cellular respiration because it's needed for the last setup of cellular respiration. So this is important because these are going to go off to another step. It generates 4 ATP by going through these steps. We have some other kinases that are going to add phosphates back onto the ADP by first removing one here, then removing another one off of here. And at the end, pyruvate kinase is the one that makes pyruvate. Pyruvate will be important for the next step. And so here you see it makes 1 ATP, it makes another, but don't forget that this happens 2 times because we started with 2 3-carbon molecules. Now that pyruvate is going to need to be prepared for entry into the citric acid cycle. So it gets oxidized in order to enter the citric acid cycle. So we have a big rearrangement of molecules going on here. Pyruvate has this carboxyl group on the end, and carboxyl groups, remember, have 2 oxygens and 1 carbon. That's CO2. So if we remove that CO2, then it can be released. That CO2 is released. And then we have something called a coenzyme A that will reduce NAD plus to NADH. So it's donating an electron to NAD to become NADH. And also that coenzyme A is going to get attached to the acetyl group that was left over on the pyruvate. And we've created something called acetyl-CoA. That acetyl-CoA is going to be then going into the citric acid cycle. So now we're into our second step, which is the citric acid cycle. And this is how we're going to break down that acetyl-CoA that was just made during the oxidation of pyruvate. And we are going to create some products and byproducts. So for the 2 acetyl-CoAs that go in, because remember there were 2 pyruvates, 2 acetyl-CoAs that go in, we're going to lose the rest of the carbon dioxide here. So 1 acetyl-CoA will release 2 carbon dioxides. We're also going to make 1 ATP, or sometimes we can make another energy molecule called GTP. Usually we're going to be going for ATP. And 3 NADHs, and 1 FADH2, which is just another electron carrier. These electron carriers, again, are going to be used in the next step of cellular respiration. They're really important for that last step. So we made a really small amount of ATP here, and we exhaled some carbon dioxide and released it, and then we're making these electron carriers. Notice how the citric acid cycle is a cycle. So for glycolysis, it was a straight pathway, but the citric acid cycle goes around and around because this very first molecule, oxaloacetate, is used to convert acetyl-CoA into citrate. And then that's going to be going around through a bunch of intermediates, and eventually it gets made again. So it can go over and over and over again rather than it having an endpoint. So that's really interesting. And another interesting thing is that these intermediates all along the way can be used in several other different pathways in our cells. You do not need to memorize all of these steps, but what you do need to be able to do is look at a step and tell me what's happening. So over here, I can see that there's something going on where NAD+ is converted to NADH. You should be able to tell me that that's a reduction reaction, that NAD+ is being reduced. You should be able to tell me that because oxidation reduction reactions are performed by dehydrogenases, that this conversion of isocitrate to alpha ketoglutarate is done by some sort of dehydrogenase. Likewise, down here, when we're generating our energy, phosphorylating something, you should be able to tell me that that's performed by a kinase. We learned that in the first video. Right, before we go on to our last step of the oxidative phosphorylation, I want to make sure we know the structure of a mitochondria. So a mitochondria has a double membrane. We talked about that when we talked about the endosymbiotic theory, that these have a double membrane probably because they used to be other prokaryotic organisms that were engulfed by another prokaryotic organism. So they ended up having two membranes. So we call that outer membrane the outer membrane, and then there's something called the inner membrane. On the inside of the inner membrane is what we call the mitochondrial matrix. So these are important because you'll see where different things are happening. The Krebs cycle that we just talked about happens in the mitochondrial matrix. If you recall that glycolysis happened in the cytoplasm of the cell. So in the mitochondrial matrix, you've got the Krebs cycle happening. Oxidative phosphorylation happens along the membrane, the inner membrane here, and it involves the space between the two membranes that we're going to call the intermembrane space. You can kind of think of this like a donut, because a donut has a hole in the middle, and hold on. I had something that I could hold up for this. No, I can't find it. I found something that I could hold up for this. So the outer part of this CD, if you remember CDs from Once Upon a Time, the outer part would be like the outer membrane of the mitochondria. Then the inner membrane is going to be the little circle on here. The middle would be where I can stick my finger through. That would be the matrix. And then between the two membranes, so all along here, that's going to be the intermembrane space. All right, so this electron transport chain is the first half of oxidative phosphorylation. So we can break it up into two processes, the electron transport chain, and then the next one is called chemiosmosis. The electron transport chain, the main idea is that it transfers electrons along a series of integral membrane proteins. These are embedded in the inner membrane of the mitochondria. So these electron carriers that were generated during glycolysis and the citric acid cycle go to the inner membrane, and they are oxidized by these proteins. So they lose their electrons. So those electrons get transferred onto the proteins, and they get transferred onto several other proteins along the way. And all along the way, as they are reducing these proteins, the proteins undergo a conformational change. So that means they change their shape, and hydrogen ions get pumped across the membrane into the intermembrane space. So that's that space between the outer membrane and the inner membrane of the mitochondria. So these are a bunch of redox reactions that happen. They oxidize those energy carriers. They reduce the proteins. The final thing that gets reduced, the final electron acceptor, is oxygen, and this actually causes the formation of water. So now what happened is that because those hydrogen ions were getting pumped across, we've created a proton gradient. So a chemical gradient has occurred where there's going to be a higher concentration of protons in the intermembrane space compared to the matrix. Now, hydrogen ions are not able to pass a membrane because they are charged. And so that what they're going to do is seek to go from high to low concentration, like we've talked about before when we talked about diffusion, but they can only go through this proton channel called ATP synthase. As they go through ATP synthase, this is facilitated diffusion because it's going across from high to low, and it is going through a helper that's facilitating it. As it goes through here, it actually causes a weird thing to happen. It's really cool if you watch it molecularly happen. Or modeled, I guess, is that it turns. So it's almost like a windmill or something, that these are almost like wind making it turn. And as it turns, it pushes phosphates onto ADP, and it makes a whole lot of ATP. It actually generates 90% of the ATP in cellular respiration, in aerobic cellular respiration. It's aerobic because oxygen had to accept that electron at the end of it. So this is all powered by that gradient that was created by the protons, which is why it's called chemiosmosis. So theoretically, we talk about how many ATP are generated, but this actually varies due to a lot of different reasons. So some of our electron carriers are a lot better at carrying electrons than others. So if we look back here, you see that this whole thing could be happening, all along the way, lots of different times. So if we have a bunch of NADH floating around, then it might be oxidized by this protein, and a bunch of FADH2, it might be as well, but it also, some other things might happen along the way, and it might not do as great of a job carrying those electrons. And actually, NAD plus is a lot better at transferring electrons than FAD plus. Interestingly, you find more of this in our liver and more of this in our brains. We also may see that some of the intermediate compounds get transferred to other pathways. So then some ATP may be not generated because it's going along a different pathway. And then also, sometimes you won't get enough protons pumped across the membrane. So if you only get a little bit of hydrogen ions pumped across the membrane, you're not going to get as many going back through that ATP synthase to generate ATP. So that's why when you see this, if you look on a bunch of different websites, if you're a person that likes to look at different videos and websites, you're going to see different amounts of ATP listed for that last step, because it's theoretical. It depends on the day, it depends on the conditions. So the glycolysis would make 2 ATP, Krebs cycle would make 2 ATP, and then the electron transport chain, that's going to help facilitate a whole lot more, 30-ish. I've seen anywhere from 30 to 34, so it depends. So this is how we get most of our ATP, and this is actually not that efficient. About 34% of the energy in glucose that we extract through is extracted through cellular respiration, and the rest of it is lost as heat. So remember, we're increasing entropy. We're constantly fighting entropy. All right, we finally made it to the end of that video.